Welcome to Mendel University, home of the world-famous genetics department, where recently major outbreaks in genetic research have occurred. Currently, Dr. D.N. Andrews and Dr. Jean Home have been conducting research in the genetic structure of humans. Their most recent discovery has the possibility to change humankind.
Through our knowledge of genetics, Dr. D.N. Andrews and I are striving to create the perfect human.
It's a delicate process that involves extensive research into DNA and pedigree. To begin the process, we had to start with the core of all genetics, DNA.
Deoxyribonucleic acid, or DNA, is the molecule that codes the genetics of all living organisms. It comes in a double helix structure and is built from a sugar phosphate backbone and nitrogen bases. During meiosis, the DNA must condense but keep its surface area to volume ratio. It does so by supercoiling around 8 histone proteins in a histone H1. Together, this is known as a nucleosome, and eventually the many nucleosomes create the chromosome. Now, the genetic loci on the chromosomes house the alleles, and alleles determine the genotype of the person, which manifests in the phenotype, or a person's physical attribute.
To make the perfect human, we need to focus on the alleles. Now, alleles can be either dominant or recessive, and they are passed from the parent to the offspring. They determine anything from eye color to hair texture, depending on which allele is dominant and recessive. For example, we want our person to have blue eyes, which is homozygous recessive. Now, to get this desired trait, we need to look at the parents and their genotypes.
To get the eye color we want, we need to make sure that the parents both have blue eyes, meaning they both have the homozygous recessive genotype. We conducted a monohybrid test with our subjects to ensure that their offspring would have the same homozygous recessive genotype, as shown in this Punnett square. Now that we have the desired phenotype, blue eyes, we can move on to pedigree.
The pedigree charts we have are the genetic family history of our subjects. We needed to double-check the genetic history of our subjects to make sure that they didn't have any genetic mutations. It's a good thing we double-checked because our original female subject was actually a carrier of sickle cell anemia. We were forced to change to a different female subject, who, as it turned out, had a perfectly clean genetic history.
Now, the way we presented our information may make you think we're going to breed our subjects. We aren't that cruel. We decided to take advantage of our amazing biotechnology equipment.
Basically, we removed a recently replicated DNA strand from our subjects using restriction enzymes, and then we replicated the DNA using polymerase chain reaction.
Polymerase chain reaction is the process of amplifying DNA to produce large quantities of a specific fragment of DNA. The DNA is inserted into a thermocycler, where the following process is repeated until the desired amount of DNA has been produced. First, the DNA is denatured. This is when it is heated in order to break the hydrogen bonds and separate the two strands of DNA. Next, it is annealed. This is when the temperature within the thermocycler is cooled in order to allow the two anneal primers to attach to the opposite ends of the target sequence. Lastly, it is elongated. This is when the enzyme DNA polymerase begins to pull in corresponding free nucleotides and attach them to the original strand to produce replicated DNA.
It took weeks, but we were actually able to amplify the DNA enough to make it clearly visible to the human eye. Who knew amplified DNA looked so much like candy?
We will get our intern, Nan Disjunction, to put our person in position and prepare the scanner. Remember, Nan, to remove anything in close proximity to the scanner. It scans everything in sight. Okay. We are very lucky at Mendel University to have the newest technology. It's basically a 3D printer, but it can turn sequences of DNA into actual organisms.
What? How is this possible? One sec. How did this happen? You idiot, that's not candy, that's DNA! She just ate DNA!
Can I go now? I have a history class to teach.
I thought we were making a biology teacher.
We were. According to our microscope set to times 500,000 magnification, there's something wrong with the nucleosomes. Wait, these aren't histones. They're histriomes.
You removed everything from the scanner, right?
Well, you guys were taking a while, so I decided I would catch up on my history homework, and I guess I may have might have left my book. Maybe.
Why, you little...
That's the entire project! This cost millions of dollars! You